Have you ever wondered how scientists see things smaller than a grain of sand? Today we explore microscopy, the science of seeing the invisible world. Microscopy is the use of microscopes to study objects too small to see with our eyes alone. It helps biologists explore cells, bacteria, and tiny structures inside them. There are two main types: the light microscope, which uses light rays, and the electron microscope, which uses beams of electrons. Each has different powers and uses. Light microscopes magnify up to 1500 times and show living, colored cells. Electron microscopes magnify over 500,000 times, but only show dead specimens in black and white. The eyepiece and objective lens magnify the image. The stage holds the slide, while the mirror reflects light through it. Coarse and fine knobs sharpen the focus. To calculate magnification, we use this formula. Magnification equals image size divided by actual size. Both measurements must be in the same units. Example 1. An image measures 50 millimeters. The actual cell is 0.5 millimeters. We divide 50 by 0.5 and get a magnification of 100 times. Example 2 is trickier. The image is 8 centimeters, actual size 40 micrometers. Convert 8 centimeters to 80,000 micrometers, then divide to get magnification of 2,000 times. Many students forget to convert units before dividing. If your image is in centimeters and the actual size in micrometers, always convert to the same unit first. Remember the mnemonic IAM. Image equals actual times magnification. Light microscopes show living color. Electron microscopes show finer detail. Always match your units. Subscribe to Ovasaabi for more lessons that make biology simple. Visit ovasaabi.com and keep exploring the amazing world of science.